Right, I'm going to talk about application of adaptive designs in clinical trials research. So how many of you have heard about adaptive designs? Do you want to raise your hand? Have you heard about adaptive designs? You've heard? Okay. All right. Okay. That'll be interesting. Right. Okay. Or maybe I can use a mic if you.
If there is a mic.
Yeah.
Yes, please.
Are you? Is it okay? No.
I don't think it's working.
All right. I'll try to speak up. Right. So I think I'll try to make sure that this session is more interactive. I don't want to spend a lot of time talking. We want all of you to contribute as well and have kind of like a discussion on things. So first of all, I think it's good for me to declare some of the things. I work for the University of Sheffield as a research fellow in medical statistics, and also I'm a leader in Adaptive Designs Consort Extension Project, which is funded by NIHR and MRC as well. No other conflict of interest to declare. So in terms of the objective of this talk, I want to highlight some of the limitations of clinical trials research at the moment, which is focusing on fixed sample size, randomized controlled trials. And also, I want to highlight how we can overcome some of the shortcomings of fixed designs, randomized controlled trials. So I'll talk about the concept of adaptive designs and also look at some of the prospective and retrospective case studies. So prospective, these are kind of like trials that have been conducted or that are still going on, which have been designed as adaptive trials. And retrospective, these are trials that have been finished. We've conducted these trials within the University of Sheffield, some of them, and we redesigned them as adaptive to show the opportunities that can be used there. So I'll also try to highlight some of the considerations on adaptive designs, and then we can have some discussions and questions as well. So what are the issues that we are having at the moment? The problem is we investigate a lot of treatments from early phase to confirmatory trials, but only maybe 1 in 5 of these treatments end up being approved in practice. So we spend a lot of time investigating interventions which are not really good for patients. So the issue is we're spending a lot of money investigating interventions that are not good enough. Secondly, what about the issue of patients? We recruit a lot of patients in these trials, but actually, first of all, they are not benefiting from these trials. So there's a balance between patients and community aspect as well. Because if I'm recruited into the trial, of course I need to contribute to evidence later on, but also I need to benefit as part of the trial. So we're also wasting a lot of resources because we can terminate some of these trials early because we know the interventions are not good enough. So another issue is about efficiency in some of the cases. One of the issues is when you are designing a trial, you make assumptions about the design. Some of you, you need to go to a statistician, have a discussion about the research question, the primary endpoint, other aspect. Then you say, okay, I need a sample size. How many patients do I need to recruit in order to address the research question? And then you're told, I need you to give me some extra information about the primary endpoint. It could be variability, or it could be event rate in the control group. So we make assumption either based on systematic review, or sometimes it's difficult because we don't have a lot of information on these parameters. So we've realized that actually, in most cases, our assumptions are not good enough, right? So you end up saying, oh, you know what, I need to conduct this trial, but I'm not sure about the variability, but this is the starting point. So you might either over-recruit participants or you under-recruit participants in a trial. The implications are obvious. If you over-recruit, it becomes an ethical issue. Why do you need to over-recruit participants in a trial? The issue about resources as well. Why do we need to spend more resources when we know that we can answer questions without over-recruitment of these participants. So I've been involved in grant applications where we review a lot of these trials. So I was looking at a case study where they wanted to investigate how physical activity interventions can help patients in some neighborhoods in the UK. So they did a systematic review, which is a really good thing, to find the gaps in the literature. So they found 6 interventions, potential interventions, which could be tested. And then they decided to just cherry pick only one of those interventions to say, okay, we think maybe this one is gonna work. We want to test that against the usual care. Just imagine, you spend 10 years running a trial with that intervention. It doesn't work. You come back to the same systematic review. You pick another intervention. You do the same, another 10 years. So we end up spending more time, right? Instead of looking at all those interventions which are competing in a single trial, right? We run a single trial for 10 years, a multiple arm. We've got an option where we can drop even interventions which are not good enough and carry over those which are promising. And there's some cases where you need to think differently, right? What we tend to do is we investigate treatment at population level, saying what is the average treatment effect? But actually what you are realizing, especially in areas like oncology, some treatments, they work for certain patients, not in general. So we need to explore what sort of patients are more likely to benefit from such treatments other than just looking at the overall population. So you might start with the whole population, then you've got subgroup within those, the whole population. You investigate and see whether the treatment is benefiting certain subgroup than the others, and drop certain subgroup as you progress in a trial. So this is called population enrichment design. And of course, as I said, it takes time to investigate a lot of these treatments as well. As you know, there are a lot of ethical issues where you want to make decisions as early as possible, especially in terms of outbreaks. So you can't start a trial and then wait for 10 years to make decisions. So we need to use the accumulating information to inform decision making as well. Right. So what is an adaptive design? The concept of fixed randomized controlled trial is highlighted here. It's very simple. We design a trial, then we know the fixed number of participants that we need to recruit. We follow these patients up until the end of the trial. After gathering all the information about the primary endpoint, we analyze all the information, then make decisions whether the treatment works or not. But it doesn't give us opportunity to look at the data, right, during the course of the trial. So for adaptive designs, we're saying, okay, hang on. We need to take some insurance, right, for unforeseeable events during the course of the trial. So we need to look at the data during the course of the trial, right, to see what's happening. Then we need to modify some aspect of the design. I will look at the aspect of the design because it could be confusing for some of you guys. Then we review, we adapt a trial, adapting or changing those aspects of the trial. Then we progress if necessary. But we need to do it in a planned manner. We can't just do it like in a kind of like a random thing to say, today, just want to look at the data and change the things as I wish, because there are implications as well. Another important aspect is about validity of the trial. We need to make sure that the inference that we conduct at the end of the trial and during that analysis is really good enough to inform decision making. So different adaptive designs, they need different statistical methods to analyze and inform decision making. But you don't need to be concerned much about what sort of statistical method. You just need to understand that there are implications. So you need to talk to your statistician. There is an issue about integrity as well. Because at the end of the day, you need to convince the research community and even regulators to say, the results that we are giving you from this trial, they are really good enough to inform decision making. So they should be comfortable with the way that you conducted your trial. Right, so I'm going to start by looking at this case study of sample size re-estimation. There is a retrospective study conducted within the University of Sheffield. It's called the RIDEFACT trial. They were evaluating a point of care cardiac marker panel in patients presenting to ED with suspected MI. So the primary endpoint was quite simple. They wanted to discharge more patients safely. So they were looking at ways to increase the discharge rate of patients. So they assumed a 50% hospital discharge rate based on some information, which was okay during that period. And they assumed that the 5% increase is clinically important to look at. And they assumed 80% power and 5% type 1 error rate. Right, using that information, a statistician would do the sample size calculation and come up with a figure: 300 and 130 patients required to be recruited in the trial. So we divide by 2 because it was a 2-arm trial. Here's the surprise. Instead of the 50%, you observed, sorry, you can't see properly, but we observed less than 25% event rate, right, in the control arm. Right. Here are the implications, because we know the maximum sample size is obtained when the event rate is 50%. So any reduction in the control event rate, which means you have recruited more participants than necessary. So I did, during my PhD, I looked at the change in the sample size as you increase participants during the course of the trial, because we know when these patients were recruited. So the first give you different approaches to estimating the event rate. Right. You don't need to worry about what method was used. And this is the sample size. This line gives you the sample size which is required, which is the fixed 330. And this is the required sample size. If you assume that you're going to do sample size re-estimation after every patient, right? But you only need to do once. So I was trying to illustrate some of the implications. So you can see that very early on, it's not really good to do sample size re-estimation very early on. You need to wait to have more information to inform decision making. So in terms of the implications,
Sorry.
Yeah.
Question.
Yeah.
Since yesterday you told us that we can interrupt, can we?
yeah. Oh yeah, exactly. You can interrupt. Yeah.
But when you did that,
Yeah.
did you also look at the ideal number of patients? So instead of doing after each and every single patient,
Yeah.
did you also make an estimation of the optimal number of patients or batches of patients that when you can redo your estimation?
Yeah.
Every 5 or 10 or whatever.
That's a really good question. We did another research before that, looking at what is the minimum sample size required to do a sample size re-estimation. So we know already for like binary endpoint, normally you need more than 100 patients to be kind of like good enough to inform your sample size re-estimation. Right. For continuous outcome, you need about more than 35 patients per group. So we've got that information already. We've got a paper published and it's being used already. So we're pretty comfortable with that approach. I was only trying to illustrate, in this case.
Because the type of one of the things perhaps to think about is also the type of outcomes that we'll be having in this kind of trials, which would probably be more likely binary.
More likely,
Oh, right. I think that's a really good point. You need to really think about what sort of endpoint are you looking at. If you are looking at binary survival,
could be just survival.
Yeah.
or progression to
To
severe disease.
severe disease or whatever. It's kind of like a different, but they kind of like approach is kind of like the same, but they've got different implications in terms of the minimum sample size that you require for that. Is that okay? Any other question? Yeah.
Yeah, I'm just trying to think about an outbreak situation.
Yeah.
Whereby you can start a study,
Yeah.
thinking it's going to be effective,
Mm.
and then the outbreak stops even before you reach the number
Mhm.
where you thought you could estimate.
Mm.
How do you go about it?
Right. This is kind of like an interesting question because there are sometimes when you start a trial, a design, you try to be optimistic about different aspects, right? But then those situations, they are more about practical aspect. You're saying, okay, I had planned this trial and I was hoping this to happen and change aspect of the design, but the outbreak has stopped. So another aspect is, are you expecting another outbreak to happen in the near future? That's another question that you need to think about here.
Yeah. So the WHO blueprint Andreas is going to talk about is looking at exactly that question. You know, how do you deal with trials that might run across multiple outbreaks? And currently we've come up, I think, with 3 different options. One is you set the trial at the start as a fixed sample size, and if the outbreak doesn't give you the sample size, you just wait. Don't look at the data, just wait for the next outbreak.
For the next outbreak,
complete
yeah.
the sample size. The second option is to have it pre-planned as an adaptation. So after the outbreak, you do a sort of blinded look at the data and see if you need to change the design. You know, do a sample size re-estimation or do other changes to it.
Yep.
And the third option is to do a sort of meta-analysis,
Yep.
where you combine data across trials. So it's not fully worked out, but there's a number of different options, one of which is an adaptive design.
Sorry, just to, maybe then we continue your presentation, but we should also consider a situation whereby there are not just cases during an outbreak. There are some of these diseases where you do have cases in between outbreaks, fewer, but you still have cases. So that should also be considered in the trial design, including the fact that during outbreaks and outside outbreaks, disease presentation may be different, and that will require further adaptation.
Yeah, there are different implications because if the population is kind of like changing from one outbreak to another, then you've got kind of like a different problem altogether. I think we're going to have another kind of like a discussion when Andrea is going to talk about that. Can we continue? Right. So if you look at that, over-recruitment, this is the median over-recruitment of 861 patients. Right. This is quite a lot, to be honest. So what happened with this study is we ran out of funds at 2,243 and went back to the funder, to NIH or HTA, to say, hang on, I think we've got a problem. We wanted to recruit that, but we couldn't because at the moment we've run out of funds because recruitment is a challenge. I think you all know that. And the funders, they say, oh, hang on. Yeah, this is a really important question. We might give you some money, but we need to understand the value for money. If your study is not being in a position to answer the research question, right, and it's going in the wrong way to say, actually, the new treatment is not good enough, what's the point for us giving more money? So they requested futility analysis by an external DMAC, independent monitoring committee, looking at what's the probability of your study giving clinically important significant result at the end of the trial, given what we have observed now under different scenarios of the remainder of the trial. So they've realized, actually, we have answered the research question already. there was no need for them to give us extra money. So they said, hang on, no, we can't give you money. Right, we need to stop the trial. And I'll look at this trial later on. I think what you need to understand here is, if we had done a sample size re-estimation during the course of the trial, there was no point for us going back to the funder. We could have known that actually, at this point, close to that point, we would have answered the research question. So we stopped the trial there. That was it. So it's just another case study to show that sample size re-estimation can help you to address some of the challenges there. Right, so this is another study which is ongoing. No, it finished, but they did a sample size re-estimation as well, and they faced a similar problem with that. They assumed the endpoint was a quick endpoint. As you know, adaptive design, sometimes you need a really quick endpoint or immediate endpoint to activate the adaptation. So they calculated the sample size, and it was 462 to achieve 90% power. Of course, you can adjust for dropout, and you're going to increase a little bit, assuming that you're going to lose a certain proportion of participants. And they did a blinded review sample size re-estimation. Blinded in the sense that you don't need to know the treatment that patients were allocated to, which is good because we don't want to introduce bias during the course of the trial. So you just look at the overall estimate using all patients. So they did that, and they realized actually they were wrong. Their assumptions were not accurate. So they had to increase the sample size by 40% to 810 in order to address their research question. At the end of the day, they've managed to show that actually the 2 doses that they were investigating were all clinically important for patients to use. So it's another case study that can be used. Now, we go back again to the RADPAC trial. This is where you want to stop trials early because you know the answer already. So in terms of outbreaks, sometimes even when there's let's suppose there's a first wave of an outbreak, you might even want to look at the data to say, hang on, for the treatment that you are kind of like investigating, do we know the answer already or not? So you can design that trial as adaptive in the sense that you want to drop certain treatments during the course of the trial. For RADPAC trial, I redesigned the trial with 2 interim analyses. So we say we want to analyze data in 2 batches. The first batch during the course of the trial, we've got all the information on those participants recruited. We need to analyze them. Second batch, we need to analyze them again, and then we do the final analysis if required. Then, as you know, there's a penalty. When you analyze data so many times, you need to account for the fact that you are doing multiple interim analyses. And your statistician will know that is going to adjust for type 1 error rate to make sure that it's below a certain level, like 5%. So we use certain stopping boundaries. So during the course of the trial, you need to sit down with your clinical colleagues, your research team, to say, okay, what sort of clinical boundaries do we want to use to stop a trial? So in this case, I used a really stringent stopping boundary to make sure that whenever we stop a trial, we've got overwhelming evidence about the treatment effect. You don't need to know about the type of boundaries or whatever, it's another issue. So, in this case, the red line, this is the observed treatment effect as we increase the number of patients. Right. This green line is the lower confidence interval of the treatment effect, and the purple is the upper confidence interval. This line is the stopping boundary to say, if you observe any treatment effect which is above that, we need to stop the treatment in favor of the one of the new treatment, which is point of care. If you observe anything that is below that, you need to stop in favor of the usual care. If you are in between those 2 purple lines, you need to stop for futility. As you can see, the results are overwhelming. Far above the stopping boundary. So basically, you can stop the trial at that point. You don't need to continue because you know the answer already. Right. And then you can try to work out in a different scenario. What are the potential benefits of doing that in terms of patients? So in this case, under different scenarios, you can look at the patient saving within the trial, and also you can look at long-term benefits as well. So it's something that you need to consider. Do you have got any questions relating to that, or any issues that you want to raise? Yeah.
I have futility. How do you decide whose condition to stop the trial about for futility?
For futility. Right. The idea is, in this case, we're saying a treatment is futile if the difference between the two treatments is close to zero. Right. It should be close to zero because that's the null hypothesis, right? But then you need to kind of like figure out the upper and lower bound to say what threshold of that upper boundary above zero that we need to make in order to say the treatment is futile. So I sat down with the chief investigator of the trial, and it gave me some clues to say, you know what, in some of these cases, I'm a bit worried if the treatment is kind of like maybe less than 1% of the, because we said the actual treatment effect is supposed to be, was it 5%? If it is less than 1% or even 0.5%, then I don't think it's benefiting patients. So we had kind of like went through kind of like a discussion. Then I had to translate that information of the treatment effect to statistical stopping boundary. So it's a discussion that you need to have as a team. It's not kind of like only a statistical issue. Yeah.
Yes, that's a question more or less similar to his.
Mm-hmm.
I just want to know, because when you design a study, you before you start, you will, in case you're intending to do interim analysis,
yeah,
you also set the stopping rules and the,
yeah,
what rule you will use for futility or for effectiveness.
Yeah.
And in case, yeah, but then I'm wondering, when do you set the adaptive assumptions? Do you wait until you do the interim analysis and see results, or you do that that is before you start the study?
Adaptive assumption. Are you talking about the timing of interim analysis or?
For example,
yeah,
you do an interim analysis,
yeah,
and then you realize you need maybe to recruit more participants to one arm than the other.
Mm-hmm.
Yes. Do you wait to make that decision at the time you do the interim analysis and seeing the results, or you can pre-plan that in advance of you starting this?
You need to pre-plan most of the adaptation process. You need to pre-plan it to say, this is what I'm going to do when I'm looking at the data. Mm-hmm. If you pre-plan, your statistician will try to factor all the kind of like planning scenarios. If that happens, this one will happen in the design of the trial. Mm-hmm. So you can work out the implications of different decision-making situation of that trial. And some of the stopping boundaries, they are kind of like flexible to say, okay, if you assume maybe you need to wait and do interim analysis at a certain point, and due to certain circumstances, you couldn't do it because maybe the outbreak is stopped, then you can look at the data at that particular point and adjust the stopping boundary. The fact that instead of waiting at 50%, you are now looking at the data at 35%. So some of the things you can work out backwards. Does that make sense?
Okay.
Yeah. Uh-huh.
Yeah, just contributing to that. I think the main point is the fact that obviously adaptive designs are kind of still new being adopted by various researchers, and for the same reason, there is still not a very clear guide on what you do. And isn't that the reason why you now have the ACE project that you're working on, which is sort of a guideline when you're doing adaptation? And therefore, what are the guidelines on how you should be able to specify the adaptation that will be involved into the trial? So that's one of the projects that Munya is involved in. To try and, as the same as you have a consult, you have an ACE which is now guidelines for adaptation, and then it pre-specify, it tells you how you should do the adaptation and how, if you're doing at the design stage, analysis stage, sample size stage, that needs to be specified with all the different options. So it would be very similar to how you do a clinical trial. But I think when you have an adaptation, then you have to pre-specify that. So then if you're going to be doing an interim analysis, I suppose that will go into your statistical analysis plan as well.
Yeah, exactly.
And so
Thanks,
you
Eric.
refer also to the document that was prepared before, one of the documents that were prepared before this meeting. There were some examples in the clinical trial module, there were some examples of different ways of going about that. But this is, I think, is perhaps would be another session to
maybe to discuss and as well. There are a lot of things to learn, and as Eric said, we're trying to kind of like improve how adaptive designs are being conducted in practice.
And we're not trying to make you all statisticians here.
No.
That's why otherwise people like Eric and Munya would go unemployed. So
Exactly.
so it's more about you knowing what your issues are and what you got to start thinking about, and then of course working with statisticians who are familiar with this type of trials.
Exactly. It's about understanding what you need to think about, and when you're having a conversation with myself and Eric, then we can have a fruitful discussion at the planning stage, and you know exactly what you're looking for. That's the thing. But I need to proceed again. There is a similar, slightly different case study, but I wanted to illustrate that you can design things differently depending on what you want as a researcher. In this trial, the researchers, they said to me, hang on, at the first interim analysis, we don't want to stop a trial for efficacy because we need to wait. If the treatment is kind of like effective, we need to wait longer to make that decisions. We want to stop early only when the treatment is not good enough at the first interim. So at this first interim, you're only allowed to stop only for futility. At the second, they say, okay, now we need two options. We can now stop the trial either for futility, if we proceeded after the first interim, and also stop for efficacy. So it's just to illustrate that you can design the trial differently depending on what you want as a researcher. So this trial, instead of the treatment being effective, the treatment was futile. So if you look at those two boundaries, as I said before, the confidence interval, the treatment effect at the second interim analysis was within that region. Right. Here is the problem that you face when you are doing this interim analysis that you need to understand. Sometimes you are faced with difficult decisions, what to do. The confidence interval, the upper limit, is on the wrong side of the boundary. So you're saying, okay, the treatment is futile, but at the same time, there's a chance that it could be on the other side. What do? This is where your statistician will help you to simulate, to try to predict what could happen under different scenario, given what happened to that point, and different scenarios for the remainder of the trial. So we did some simulation, simulated about 100,000 trials. Right. And we say, okay, let's assume that the treatment is as observed. Right? The probability of failing to reject the null hypothesis was 99.7%. So you're kind of like sure to say, you know what, this is just futile. I don't think we're going to show the effectiveness anyway. What's the point of carrying forward? Then clinical investigator said to me, okay, what about if you assume we've observed it almost close to zero, but the remainder of the trial is close to the maximum that we assumed at the design stage, which is 6%. And we did that simulation and got sort of like maybe 15.4% chance. And after a long discussion, they said, you know what? We know we observed close to zero. Why do we expect the remaining 35% of the patients to have a treatment effect of 6%? Even if they have, the chances of observing a clinically meaningful at the end of the trial is just 15%. So they had said, you know what? I think we know what to do. We stopped the trial. We analyzed the data, accounting for probability, the chance of stopping early, and interim analysis, so using different methods. And the results were consistent with those observed at the end of the trial. So it's just to illustrate that you can do things differently. Any questions? Yeah?
So again, thinking about how to do things differently, so when you were doing that, you had predefined a certain alpha and a certain beta.
Yeah.
Okay. So in your simulations, do you also start playing with the type 1 and type 2 error, depending on the clinical conditions? So my question essentially is, what's also important, this is, of course, it is a statistical question, but it is also a clinical question behind that. And depending on the severity of the condition, the alternative options that are available to treat that condition, you might wish to play a bit with the type 1 and the type 2 errors. So to be, you might be inclined to err on the side of accepting something that, or rejecting something that may or may not be effective, no?
Yeah, that's a really good point. There are a lot of consideration that you need to look at when you are making those decisions. Like in this case, we're looking at treatments which are some treatments that are being offered already in practice, but we don't have evidence for them. Right, so this is an example. Sometimes you're looking at a treatment which is not even available in practice, and the condition is kind of like critical, severe. You've got a different things to consider. So you can play around with the chances of making a false positive to say, okay, do I need to take a risk to say there is no treatment which is out there, right? What sort of risk can I take to allow maybe a wrong decision of taking that treatment into practice? So you can do that during simulation depending on the clinical question to say, okay, what about if I lower my chances of false positive or increase the chance of false positive? So it's something that you need to really think about at the design stage and have a conversation around that. Is that okay? Any? Yeah?
I've seen in the two case scenarios that there were two interim analyses.
Yeah.
And the one is for 50 and 75, and for this one is for 50 and 50, 65. What is the reason behind to choose this percentage? That's purely clinical, or is it statistical that we have to determine before we do the...
Yeah, that's a really good question. I'll try by going through why we used 50%. The reason why we used 50% is because, as I said already before, looking at that graph of the sample size, we know the regions where variability is too high. The uncertainty is too high to make decisions. If you look at that graph, we don't want to look at the data in that region, because we know the uncertainty is too high. We know that already. We've done a lot of research on that. So areas like 50% or whatever, you could see even after maybe 40%, the results were kind of like stable for you to make decisions. So that one it's easier. You can have a conversation with your clinical colleagues. The second one, it was a balance between what is the benefit of stopping a trial early at different time points, right? And also the resources that you need to do interim analysis, right? sometimes you don't want just to do interim analysis for the sake of doing it. If you know that the benefits are tiny, right, you could do it at 85 or 90%, but then you say, hang on, we had only, there was only 10%, maybe patients, to recruit anyway. What is the point for us to stop a trial? So in this case, we had to work out what are the benefits of doing interim analysis at different time points with respect to patient serving, with respect to kind of like resources, and also our ability to do it at different points. So there's no rule to tell you that 65 is the only kind of like a figure that you need to go for. You just need to think about it carefully in terms of the balance between doing interim analysis and also some of the practical issues that you're required to do. And some of the things, some say it's about the research question, right? If you are looking at like critical conditions like maybe Ebola, I don't mind doing interim analysis after every patient. Because it's a critical condition. I need to help my colleagues to make decisions, right? These are interventions which are being used already in practice, right? So I'm kind of like flexible because the situation is different. Does that make sense? Any other question?
If you do that,
yeah,
don't you increase the chances of type 1 doing analysis every other patient?
Right, exactly. As some colleagues say, it's about the risk to say, given my research question, what is the risk am I in a position to take? Right. If you are looking at cases like Ebola, someone would tell you to say, hang on, because there's no kind of like maybe an effective treatment already, I'm willing to take and increase the chance of making a false positive on a treatment than to try to be stringent when we know that there's nothing out there. So these are kind of like, even regulators, sometimes they're kind of like flexible in terms of how they view things as well. Sometimes it depends on the population as well. If you've got a fixed population, like in red conditions where you're looking at 50 patients, what do? You are stuck. You can't do it after 200 patients because you've got only a maximum of 50. So there are a lot of things to kind of like consider at the beginning. Yeah. Happy? Can continue? Right. Right. This is an example of. Sometimes you want to investigate multiple treatments in a trial, and this is just an example which is going on within the UK. They are looking at phase 2. It's a phase 2 trial. This is like 3 doses of a drug, 20 milligram, 40, and 80, against a control in reducing insulin resistance in HIV patients. Right. So they had. Normally in phase 2, it's difficult for you to come up with a dose to say a 30 milligram is the good dose or whatever. Because as you increase the dose, you expect effectiveness to increase. But you expect also the risk of adverse events to also what? Increase. So you're kind of like in the middle of trying to optimize effectiveness and also safety. Right. The lower the dose, of course, it's safer, but it's not good enough. So they decided to say, okay, hang on, instead of taking one dose to phase 3, why can't we just investigate different doses in phase 2 and try to look at, efficacy and safety during the course of the trial? So they did that, and then 50% of the blind sample size, they had to drop 2 doses, the lower doses, because they were not effective as well. So it's just to illustrate that you can do different options when you're designing a trial as well. For more information, I've got reference. So, and time, please. Do you want to keep us the time?
Yeah, it is 10 o'clock.
10 o'clock.
Yeah.
Okay, so I can continue a little. That's fine. This is an illustration of different designs that you can look at. In this case, the researchers, they're kind of like thinking to say, hang on, we don't want to compare it to usual care in phase 2. We just want to compare different like drugs or treatments at early phase and choose which ones are promising. Then in phase 3, now we're bringing in the control, the usual care, to compare it against. Right. And the design, the good thing is like, you don't need to wait between the 2 phases. Right. After phase 2, what we do is we sit down, discuss results, publish results. We go back to the funders, to regulators, to say, now we want to start another study. You spend 2 years, 3 years, or 4 years doing that process. But you can only use a single protocol, just like there, and design a trial that combines 2 phases together, and then you work out the operational characteristics of the study. And then you can conduct the study. Yeah, you've got any questions? Yeah.
In an outbreak situation like Ebola, where a new product is under trial, and then in addition to an old product, the interim analysis is really relevant in that context.
Sorry, do you want to repeat again? Sorry.
Ebola outbreak, and then a new product is under trial, in addition to an old product, and you want to do the interim analysis. I want to see whether it's really relevant in that context.
Yeah. I don't see any problems why you can't do it. What you just need to do is to think about these things at the planning stage. When you are planning a trial, to say, okay, there are options for me to do these adaptations. Then you factor them into the design thing. There's some cases even you can bring in a new treatment, right? Because we know, like during Ebola, some companies were developing different treatments. If you can have an agreement with different sponsors, whatever, to say, okay, hang on, we've got a treatment, and there's a trial going on, and we're happy for you guys, instead of starting another trial, we can include that treatment during that trial. That's fine, but of course there are statistical implications that I don't want to spend time looking at, but it's practical to do that.
Would the previous slide you had?
Is it that?
Yeah. No, that one.
Sorry.
That and the following one.
Yeah.
What is the difference?
The difference is, this one is just looking at phase 2. This is a single phase 2 trial, where you are looking at efficacy of the treatment. The second one, and also the comparator, is just the control, is just a single comparator. The second one, these are 2 phases. The first one, you can look at it as phase 2. You're looking at efficacy of the treatment. And the comparator here is the kind of, you can say, lower dose or something like that. So you've got a different comparator. The second bit is confirmatory, like phase 3 trial. But the comparator now you're bringing in a new treatment, which is usual care.
Okay, but I could have turned the previous slide into this.
You
could have planned it as face to face.
Exactly, you could have done that. Yeah, absolutely. Right. Okay, fine. This is the last slide, and then we can have maybe discussion or something, or maybe a break. Sometimes you want to, what we do in most trials is we randomize participants using what we call equal allocation. So we say, we're going to randomize you to treatment A, treatment B, with a ratio of 1 to 1. So the probability of me entering the trial and get treatment A is 50%, and the other one is 50%. But there are cases where you might say, hang on, I think I want to increase the chances of allocating patients to a treatment that is promising. Right. It's an ethical issue. You're trying to balance a patient and society. If you know that another treatment is doing well in the trial, and you keep the randomization 50-50, when the condition is kind of like critical, then some people might be worried to say, hang on, we know that there's a treatment which is kind of like doing well, and you're keeping this patient, the probability the same. why not trying to increase the chances of allocating incoming patients to that treatment which is doing well with higher probability? Of course, there are issues. There's no free lunch. There are issues. In this trial, they were looking at three regimes. This is like oncology, right? It's a difficult area. There is no kind of like treatments. In some cases, treatments are there. So they wanted to increase the chances of allocating patients to a promising treatment. So what they did is they started with one-to-one randomization. Then they looked at the data after every patient and tried to change the allocation probability depending on the results. But they kept the probability of the control arm fixed. And the control arm fixed. Right, so what they did is the first bit, you can see like the area, shaded area. It's kind of like the probability of allocating patients to different treatments. And you can see like the TI treatment group? The probability went down. Right, went down and it was close to 0. Maybe after 24 patients, and they decided to stop that treatment because the results were not promising at all. They stopped that treatment arm. And in this case, you can see the increased probability of allocating to that treatment, but it dropped after the 24th patients, and they decided to stop the treatment arm as well. But here are the challenges that you need to think about. Are you recruiting the same patient population during the course of the trial? This is where it becomes an issue to say, are we comparing like with like? So some of these things, it really depends with the condition, and if you have got an understanding of the condition, it will be quite helpful. Another issue as well is, we know already that region is where uncertainty is quite high. So it could be because one patient had a good, good response, and that will affect the randomization a lot. So some would say, okay, why can't you fix the randomization at 1-to-1 until a certain point, maybe after X number of patients, and then you change the randomization after that? That could be another solution. So they looked at the probability of observing results, right? In this case, they will look, what is the probability of success in the final treatment compared to the control in the IA treatment group. And they found that actually the probability was just like 55% of the patients would have a successful treatment there. And they wanted to work out, say, okay, given what we observed, what is the chance of success of the TA to be inferior compared to IA? And it was 70%. This is not surprising because we know already 55% of the patients had a good response rate. So it's something that you need to consider, but there are challenges. We're still trying to work out the inferential properties of the design. That's the challenge at the moment. So there are guys who are trying to work out what do you do during analysis, because there are a lot of issues. But I think in the next maybe 2 years we'll be fine, I think. So I think I don't want to spend a lot of time talking. Do you have got any questions relating to that design?
It's not clear to me.
It's not clear, right? Right. Well, I think the basic thing is like you want to change the randomization based on the incoming results and increase the chances of allocating incoming patients to treatments which are performing better. Right. So in this case, the treatment which was performing better was the IA treatment, which is the control. Right. So you can see the probability of the TI was going down because patients were not responding to treatment. So they wanted to lower the chances of allocating incoming patients to that treatment arm. And because it wasn't performing well, the probability was close to 0 after 24 patients. So they had to stop that treatment arm to say, actually, the probability is 0. There is no kind of like good chance for us to allocate new treatment to that arm because it's not performing well. So it's just, the principle is just the same. Of course.
And the analysis is made after randomized.
Right. In this case, they looked at it after every patient because of the treatment and the time. I think the time was after every 49 days was the primary endpoint. So they were looking at, data as it comes. Yeah. So there are things that you need to think about feasibility of doing it after every patient, or you can do it after every 5 patients, or you can do it after every 10. So it's up to you to work out which approach you want to take. Yeah.
So maybe just to, again, to try and answer your question here. One element is, of course, how soon you can make that decision. So in this case, they had to wait for 40 plus days before they could decide whether a treatment was a success or a failure. If you take the case of Ebola, that time will be much shorter. It will be just a few days.
Just a few days.
Or maximum 2 weeks. And that would facilitate making that sort of decisions. Now again, if you take Ebola as an example, think of the situation with Ebola where you only had standard treatment just through it. And then you have a new drug that you want to test. So if you use that sort of design, you know that you're not happy with the standard treatment, of course, and you want to minimize the risks of people being treated with something that doesn't work. So if your drug that you're testing is promising, you want to have more patients on the drug than patients on the standard treatment. And that sort of design will allow you essentially to do that, whereby you are at the same time dealing with the ethical issue of not offering something that is not going to work, but also clinically you accumulate more data on the drug that you believe might be working, and you would know more about how that drug works. So essentially, this is what's behind making that sort of conclusion, that sort of design.
Exactly. Yeah. Another question.
Does it mean that the response adaptive randomization is limited to phase 2 and phase 3 trials, since it's looking at response? Can't we apply it in other trials, like phase 1 trials?
Phase 1, I think it's possible. But phase 1, it really depends on. Right, this is where I think the way classification could be difficult. Like phase 1, if you are looking at pharmacokinetic properties of a drug, whatever, sometimes you're not comparing two treatments. Right. So you can't use that because the research question is kind of like different. Yeah, but like pharma, they classify like phase 2 into phase 3, 2A and 2B. But in those ones, because you're comparing to something else, you can use it. Yeah.
Yeah, just in response to adaptive randomization, one of the assumptions is that the patient population is homogeneous across the duration of the trial. And I think the big caution with outbreaks is that often that's not the case, because early on in an outbreak, you only see the severe cases, and then you implement public health measures, and you go out looking for cases and doing contact tracing, and you start to see the milder cases. So often the severity of the disease appears to reduce during an outbreak because you're looking for the mild cases. And that's a problem for adaptive randomization because your patient population is
kind of like changing
across the duration. So I think it's a design that could be a problem in many outbreaks.
Yeah, it could be a problem, I think, and at the moment even,
And if you remember that the Côte d'Ivoire, the group, we did discuss changes in in severity and response to standard treatment or LASA at the first workshop in Entebbe.
Yeah, it would be interesting from methodological perspective to say, can we account for changing in population to also influence the randomization? You know, that's another something that maybe some clever people can
I think as you were saying,
maybe try to work out.
you can probably adapt for it all, but then the inference becomes very difficult.
Exactly, yeah.
As to how you interpret,
How you
the results
interpret
when
them.
you've had multiple levels
Yeah,
of
yeah,
adjustment.
exactly. It's a similar thing with the group sequential designs. You know, the assumption of homogeneity of the population applies as well. If you've got interim analysis where you're looking at severe cases and the second you're looking at different cases, then interpretation is gonna be challenging. So it's something that you need to really think about carefully. Yeah, I think that's it for now. I think there are a lot of things to talk about adaptive designs, and I wanted just to highlight opportunities that are kind of like available. I even talked about some of the issues like population enrichment and so, etc. And of course, as I said, there is no free lunch. There are a lot of things to kind of like consider at the design stage. Sit down with all the key stakeholders. If you are involving regulators, sit down with regulators and think about your design carefully before you kind of like implement that design. And I think, okay, I want to thank Philip, because I took some stuff because we're working on a certain paper, so I want to thank that. So the discussion, do we want to do it after the break or?
We've got a scheduled break at 11.
Yeah.
Coffee.
Okay.
We can take kind of we can turn the lights on and maybe
All right.
have a chance to.
Yeah.
Stop the recording as well.
Exactly.